So now we come to summarize what I've told you so far about X chromosome inactivation and the stages. You remember this slide from before, and I've just now added some more details. We start out with the embryonic stem cells, or the cells of the inner cell mass, and watch differentiation as it occurs over the course of about 10 days in vitro. The very early stages are all about controlling which chromosome will express exist. We know that the pluripotency factors are involved in repressing exist expression, and that as differentiation is stimulated, the pluripotency factors will decrease. This will allow the upregulation of RNF12, and RNF12 will then be involved in this counting process by its own expression. We know that X chromosome pairing, this transient chromosome kissing that occurs, somehow relates to the choice of which X chromosome will go on to be inactive, and that SATB1 and SATB2 are required at around this time point in order to bring about that silent nuclear compartment. So once exist begins to be expressed, we know that it's rapidly we know that RNA polymerase II is rapidly removed from the region where exist is found, and we have silencing of repeats and the establishment of a silent nuclear compartment. We rapidly lose the active histone marks, then exist is involved in recruiting PRC1 and PRC2 and possibly other epigenetic modifiers at this time, and we get the accumulation of the repressive histone marks. During this time, we know that there's progressive recruitment into this silent nuclear compartment, and this is why genes are progressively silenced over time. So not all genes are silenced at the same time point, but rather it depends when they are recruited into that silent nuclear compartment in terms of when they will actually be silenced. It's at this stage when we really switch to the maintenance phase of X chromosome inactivation. And one of the things I haven't mentioned to date is that one of the features of the X chromosome is that it undergoes late replication timing. And this is true not only for X inactivation, not only for the heterochromatin found on the inactive X, I guess, but also for other heterochromatin. So I'll just spend a moment to explain that. So within the S phase, we know that chromatin can be, or the DNA can be replicated at many different stages. It could be replicated early or late during S phase. And so there can be a temporal segregation of the replication of euchromatin, so open chromatin or active chromatin, compared with heterochromatin like the inactive X. And so what we know is that this probably relates to the transfer, or the transfer of the epigenetic marks from one nucleosome to the daughter nucleosome. So if we want to have mitotic heritability, we need to have that these epigenetic marks can be translated from one nucleosome to the second nucleosome. Just like with DNA methylation, you need to have the daughter strand of DNA being methylated just like the parental strand. So it's only during late S phase when the epigenetic modifier complexes that are involved in laying down repressive histone marks are actually located at that replication fork. And they need to be there at the replication fork so they can recognize their own epigenetic mark on the parental nucleosomes or on the parental histones and lay them down on the new histones being brought into the new strand that's being created. So then it makes sense that heterochromatin tends to be replicated very late in S phase, whereas euchromatin is replicated early in S phase. So this is true for heterochromatin in general, and is true for the inactive X chromosome. It undergoes late replication timing. So after the late replication timing happens, we know that we also have the association of other chromatin proteins or histone variants on the inactive X chromosome. So in fact, although you wouldn't necessarily expect it, some of these are also exist dependent. So we know macro-H2A, this histone variant associated with the inactive X, its association is dependent on exist, as is SMOOCH-D1's. SAF-A is a protein that I haven't talked about, but it is involved in stabilizing exist on the inactive X chromosome. Very late in X inactivation, we know that ATRX, which is a chromatin remodeler, is also associated, which is presumably involved in densely packaging down that inactive X chromosome. And so almost all of the features, or probably all of the features that I told you about epigenetic control in weeks 1 and 2 are demonstrated by this X inactivation process. So DNA methylation, I mentioned, tends to happen very late. It does tend to happen at this very late stage at around 10 days post-differentiation in embryonic stem cells. However, there are some CPG islands, some promoters of genes subject to X inactivation, which are methylated relatively early in this process. So like gene silencing, DNA methylation tends to happen over an extended period of time, but it is predominantly happening at this end stage as one of the last events in X inactivation. So this slide is an extremely busy slide, and so it would seem that we know a huge amount how X inactivation occurs. We know much about the molecular mechanisms. But actually that's not true. While we know many of the marks that are associated with the inactive X chromosome, we don't know exactly how they fit together. So say for example for polycomb repressive complex 1 and 2, these lay down the marks that are associated with inactive chromatin, and they're recruited by EXIST. However, we know that even when they are recruited, they're insufficient to actually silence the inactive X chromosome. And so this is actually, at the moment, is kind of where we're at. We know many of the hallmarks of the inactive X chromosome, but we don't really know how it brings about transcriptional silencing. So I just want to touch on very briefly one of the things that my lab does. So I've mentioned before that one of the things that my lab's interested in doing is using microRNAs directed against the genes that we think might be involved in epigenetic control, all of the potential epigenetic modifiers, to be able to sort out which epigenetic modifiers are important and when. So in a way, these are the potential epigenetic modifiers, this huge pile of puzzle pieces, and each in turn we can reduce the expression of each of these in turn using microRNAs directed against each one. One of the systems that we use to study actually is X inactivation. So we study this in the lab hoping to find new epigenetic modifiers that are involved in X inactivation, because we believe that perhaps one of the reasons we don't really understand the precise molecular mechanisms yet is because we don't know about all of the puzzle pieces. We don't have all of the players. The way that we do this, we need to is to use fluorophores, and I'll explain why. To be able to perform a screen, we need to be able to very rapidly assess X inactivation. And so I'll just mention exactly how we do this. And so what we've done is we've made fluorescent proteins that tag the expression from each X chromosome. So here, the paternal X chromosome, XP, is green, whereas XM, so the maternal X chromosome, produces a red fluorescent protein. So in embryonic stem cells or cells of the inner cell mass or primordial germ cells, you'll have two active X chromosomes and the cells will be green and red. However, in differentiated cells, so somatic cells of the female, adult cells, then you'll find that the cells are either red or green, and that's because of random X inactivation. So what we can do is we can use this system to be able to screen for factors involved in these many processes, these many stages of X inactivation I've mentioned. So we can take the embryonic stem cells that are both red and green, we can induce them to differentiate and watch them as they turn either red or green, but not both. And as we introduce these microRNAs to reduce the expression of each of the particular genes we're interested in, about 1,000 genes, we can study whether the phases of initiation and establishment or maintenance of X inactivation occur normally or are in some way disturbed. And this will give us some indication as to whether the gene that we reduce the expression of with microRNAs has any role in this fundamental process of X inactivation. So the reason we study this is for the same reason I wanted to tell you all about X inactivation, and that's because everything that we seem to learn about X inactivation is in fact true for epigenetic control throughout the genome. So in summary then of what I've told you about X inactivation over the past several lectures, we know that X inactivation is initiated by EXIST, this long non-coding RNA that is absolutely critical for X inactivation to occur. This long non-coding RNA EXIST coats the inactive X chromosome in cis, so from the same chromosome from which it's expressed. We know that the X inactivation centers and the surrounding X chromosome pairing regions briefly kiss, they transiently co-localize, and this influences the choice of which X chromosome will actually go on to express EXIST and become inactive. EXIST can then create a silent nuclear compartment by excluding RNA polymerase II, and we get repeats being silenced first, followed by genes. And it's by the genes being pulled into this silent nuclear compartment that they are actually silenced. And finally, we know that EXIST can recruit various epigenetic modifiers, histone variants, chromatin proteins like SMOOCH-D1 and chromatin remodelers, potentially even chromatin remodelers, and this results in the progressive layering of these redundant epigenetic marks that are mitotically heritable. and so therefore we lock in that silent state and we make sure that it's maintained for the lifetime of that organism. So in the next lecture, we're going to spend just a very brief amount of time thinking about how dosage compensation happens in lower organisms, in worms and flies.